Thank you. Welcome, everyone. Today we will have the fourth webinar for the Tetris educational series. Thank you, Sara Gutiérrez, for giving us this talk and sharing with us her expertise on genetics and genetic biomarkers, especially for radiotherapy outcomes. Sara Gutiérrez leads the Hereditary Cancer Genetic Group in Vall Institute of Oncology in Barcelona. And in Tetris, she is leading the work package on genetics. Thank you, Sara.
Thank you. So what a day. Thank you very much for having me here today. I'm very happy to do our fourth Tetris educational webinar in the context of the Tetris Young Research Week here in Milano. So I will do my best to do an introduction to genomic biomarkers in research in radiation oncology. So, okay. So the content of my seminar will be to a brief summary of the genetic susceptibility, the rationale why we thought that one of the factors that can explain the genetic susceptibility to develop side effects after radiotherapy. Then Tiziana asked me go to the basics to explain what are the single nucleotide variants and the famous single nucleotide polymorphisms that we said popularly as like SNPs. And I will do an introduction to the genome-wide association studies, and also an introduction to the polygenetic course. And then I will briefly review what work are we doing in the work package 4 in the Tetris project. So for this audience, is already known that the radiotherapy plays an important role in breast cancer treatment, not only in breast cancer treatment, in all treatments for curing the cancer. So around 50% of the patients diagnosed with cancer will receive some form of radiotherapy, and has been proved, and many clinical trials have demonstrated that the radiotherapy decrease the local recurrence and to have a beneficial effect on survival. However, there is, let's say, a small percentage of patients, depending on the tumor site, but we can say roughly around 20% of the patients can suffer or develop side effects after radiotherapy. That the most relevant for the quality of life of the patients are the late and chronic side effects. And in breast cancer, we can have after 15 years, 10 years, an increase of secondary malignancies due to the irradiation of normal tissues. We can have after 2 to 5 the development of skin effects, and also between 10 and 15 years, we can have an increase of cardiac complications. So the clinical factors of the patients and the dosimetry and other conditions of the tumor cannot explain 100% of this variation in sensitivity of the patients after receiving radiotherapy, and has been postulated many years ago, that the genetic individual susceptibility to radiation can have a role and can be another predictor that complement this picture of individual susceptibility to suffer side effects after radiotherapy. So this is, I like this picture is from a recent review in New England Journal of Medicine, explaining in a broad aspects, the radiobiological process in normal tissue. So we can understand why the DNA can have a role, because after the radiation, the DNA directly or indirectly is damaged and can have two effects: cell death or cell damage, and the cell can survive and can enter in different stages. But one is named cell senescence, and if it's inflammation, and if this cell doesn't die, can be the initial of a side effects, inflammation, and can the cell, the tissue cannot be healed and can be placed organ dysfunction, fibrosis, vascular injury, ulceration, and secondary cancers. Okay. Also, one of the evidence that we have that the genetic susceptibility can have an important role in the radiation sensitivity is because we know since many, many years ago, there are severe disease in individuals that inherited, unfortunately, pathogenic variants in their DNA that inherit from the dad and the mom. And after this severe disease, these syndromes, if they receive radiotherapy or they are exposed to ionized radiation, they have severe effects. So, for example, ataxia-telangiectasia is a syndrome due to pathogenic variants in the gene called ATM. That is a protein that encodes an important protein in responding damage after ionizing radiation. So, for example, also TP53 is another very well-known gene that if the individual unfortunately has one pathogenic variant in one of the chromosomes, so they suffer of the Li-Fraumeni syndrome, and if they are treated with radiotherapy, they can have serious effects. So there are other genes, but the presence of this this severe disease is very rare. Means that there are few individuals, so cannot explain the 10 or 20% of patients that can suffer complications after radiotherapy. I like this graph, not only to explain radiotherapy side effects, but the majority of the genetic landscape of the majority of disease, where pathogenic variants, very few frequent, very rare in the population, can have a severe effect. So this is the case for the ataxia-telangiectasia, as I said to you, the TP53 Li syndrome and Li-Fraumeni and other syndromes, Nitschmann-Bricage syndrome, for example, and they are located in this graph with a high effect. The pathogenic variants will have a high effect in the action of the protein and causing this severe disease, but they are very rare in the population. But the 20% of the patients that can have severe or chronic effects after radiotherapy are more than rare, are around 20%. And they are maybe located here in this graph, that they are maybe due to common variants, meaning that they are frequent in population, and each of these variants can have a very low effect, but together can have an effect, not only in the phenotype that is our interest of radiotherapy side effects, but also another similar complex disease, because we call complex disease that depend on the combination of these genetic variants with low effect. For example, sporadic cancer, diabetes, Alzheimer, bipolar syndrome, and not only disorders, but also normal characteristics, body mass index, height, and so on. Okay. So the goal of the radiogenomics from 15, 20 years ago, looking for the genetic susceptibility behind the radiotherapy side effects, is to have identified all these genes and to before radiotherapy to intervene the patients together with the other characteristics of the dosimetric and conditions of the patients, comorbidities, and so on, and to prevent the appearance of the side effects. So this is the goal of the precision radiotherapy through germline genetic predictors of in radiation response. But we have learned a lot in this journey, and we have assays because the response to radiation depends on the cell. So we can detect some patients that are prone to develop side effects using cell assays, blood assays, fibroblast assays. We have learned that we have identified some genetic variants that are behind these effects. And also we are learning now that there is another component of the of the DNA and RNA expression of this gene that we can also identify individuals susceptible to develop side effects, measuring RNA expression, the expression of this gene. But so far, there is not any genetic guided planned radiotherapy, okay, because there is a lack of validation studies and clinical trials in the real life of the patient. So let's go to the beginning. What are the single nucleotide variants and what are the single nucleotide polymorphisms? So the broad concept is a variant, a genetic variant. The most frequent in the DNA is this single nucleotide variant. That is the substitution of one nucleotide by another nucleotide. So you know that the DNA is packed in the nucleus of the cell, and depending on the cell phase, this DNA is really, really tight, packed. But when the cell prepares to division, the DNA needs to be not packed, it needs to be divided in chromosomes, because otherwise it would be very difficult to share the DNA between the two cells, two daughter cells, okay? So this is the chromosomes. So the human species has 23 pairs of chromosomes. One is from your dad, and the other is from your mom. Okay, this is you. So we have 23 pairs of chromosomes. One is your mom, one is from your dad. So at genetic variants, the most frequent in the DNA is this kind of variants, where a nucleotide is changed by another nucleotide, and these positions change between different individuals. These are the single nucleotide variants. This is the simple variant. There are all another type of variants, but deletions, insertions, but these are the most frequent. So the polymorphism, the single nucleotide polymorphism is the substitution of a nucleotide by another nucleotide, but they are present in the population in higher than 1%. That's why is the word polymorphism. That means it's a substitution of one nucleotide by another nucleotide, but there is this change is frequent, is present in more than 1% of the population. And is the type of variant that is used in genome-wide association studies, by definition. Okay. This is an example that I took from the genome-wide association study from Handu, Harkeran, and collaborators in breast cancer patients. One of the SNPs, this T, is exactly in the same position, the same substitution is. In that position, there is a cytosine, but the T allele that is present in 5% of the population gives a small risk to develop side effects that was identified in that study. Okay? And if you go in the genetic data sets, you find that the T allele, that is less frequent, the majority of the population has cytosine in that position, but there is around 5% of the population that has this T. This innate. Okay. So you and I, and between you, the genome is almost identical. More than 90.6% is similar. And the difference between you and me and between you is only 0.4%. So this 0.4% can have, for some people, very rare, luckily, but unfortunately for them, but pathogenic variants that can have a severe disease, a severe phenotype. For example, this individual has inherited from mom and from dad a pathogenic variant in this gene of cystic fibrosis and developed cystic fibrosis. But this is very rare. But there are other traits and phenotypes that depends not in only the combination of two chromosomes, two different variants, but the combination of multiple variants. These are the complex disease, and this is the scenario that we have learned so far that are behind radiotherapy side effects for the majority of tumors. So here in the graph is for the coronary artery disease, that a person has different variants scattered through the genome that together gives you a predisposition to develop coronary artery disease. So the genome-wide association studies. So let's go back to this graph for looking for this rare allele causing that we call Mendelian disease, that is a monogenic depending on one gene. There are different kind of studies compared to genome. It's nothing to do with genome-wide association studies. It's a family studies, linkage analysis, that is another chapter of explanation that I will not address today. And then for looking for the identifying the genetic variants behind complex diseases or complex traits or the hate or normal conditions, we need to do the whole genome or wide association study. Okay? So to answer in our interest in Tetris and required for precise and Tetris project, do common genetic variants, SNPs, influence radiotherapy side effects? So GWAS is the standard approach for answering this question. So this is very simple. A simplification or oversimplification of the process is compare the genetic variants that are in the controls, non-sampling the traits of interest that we are studying, compared to cases that have the disease or they are having diabetes or Alzheimer or so on. So we compare statistically the frequency of the variant between the cases and controls, and do different analysis to show what are these differences. So with more detail, we identify the cases and controls. We genotype. That means because the genotype is these two positions, in the allele of the mom and dad, that I show you that we have cytosine and thymine. This is a genotype. We call genotype. And one of these, each of these two nucleotides is an allele. Okay. So we use a technique for identifying the genetic variants across all genome. In many years ago, and so far, we use a technique specifically not to sequence the whole genome. Now we are sequencing the whole genome. So this strategy that I explained to you, maybe in 10 years, won't be used, because now we are sequencing all genome. It's cheaper, it's computational demanding to store the whole genomes, but these association studies that I will explain you will be done with whole genome sequencing in the future. And in fact, for UK Biobank, that they are sequenced 100,000 of genomes, they are already published whole genome or wide association studies using whole genome sequence. But in our required, precise, and Tetris, we are working with genotyping techniques that only interrogate specific positions across the entire genome. Okay. And then there is a quality control for grouping ethnicity, the different cases and controls, because the genetic variants also vary according to the ethnicity group. So, and then based on these genetic markers that we really genotyping, we can infer statistically other positions to gain more genetic variants to include in analysis. This process is called imputation, and is based because we inherited segments, blocks of DNA in the same fashion between different generations. I'm not going to enter in detail because this is due to the linkage disequilibrium, recombination, and so on. But this is not the moment to explain. But for you is to keep in mind that this is statistically inference based on reference panels of whole genome sequence that we had. For example, we have the reference panel called 1,000 genomes, TopMed. Now we have UK Biobank. And if you analyze the blocks, the segments of DNA, there are segments of DNA that are inherited in the same blocks, inherited between generations in the same, without changes, okay? So there are different, we call these different blocks of DNA haplotypes. So there are different haplotypes. So if you know 2 or 3 genetics positions, variants, you can infer the others with a statistical probability, okay? This is imputation. So from having 500,000 SNPs, you can have 20 million of SNPs that you are not really genotyping of sequence of identifying, okay? And then they do the statistical association. And meta-analysis and replication, that is very important. And also another important step in this analysis to do a post-GWAS analysis, looking for the really causing variants, because with this technique, we are not having information what is the cause, because maybe these genetic markers are linking and pointing to another positions of the genome. And in the majority of times has been then demonstrated that this list of SNPs that are statistically associated with your tree are not the causing variants, are linked to another variants that are in another region of the genome that is causing the disease. So this post-G1 analysis is relevant. So the genome-wide association studies is the estimate the effect size of individual genetic variants associated with a phenotype. So we are comparing, let's say, 6 million of SNPs versus another 6 million of SNPs in cases and controls. And for each of the SNPs, you can have the weight, the difference, the P level, and identify which is the risk allele. Because we have 2 alleles, we need to identify which of the 2 are associated or not with the phenotype of interest. And there is a significant threshold that needs to be applied because multiple comparison, and we use usually the von Ferroni correction. Okay, and according to the number of SNPs, the threshold to say that is really significant is this 5 to 10 minus 8. And this is one of the outcomes, graphical outcomes, that we usually have in genome-wide association studies. In the x-axis, we have the chromosomes, and in the y-axis, we have the logarithm of the p-value. We place the threshold that we established as after applying this correction for multiple comparison of 5 to 10 minus 8, and all SNPs up to this level are statistically associated with your trait of interest. And this also is relevant from the GWAS. Normally, we have these tables of summary statistics in which have the position of the change, the genetic variant, the chromosome, the position of the ID, the chromosome, the position of that change in the chromosome, the other allele or the reference allele that is not related with the trait, and the effect allele that is related with the if it's genetically statistically associated. MAF is the minor allele frequency that we call regarding to the reference map that we have. The allele that is less frequent we call minor allele, and this is the frequency in the general population. Okay, not needed that the less frequent allele is automatically linked to disease. It's not the majority of the cases. It's not the case. It's not that. And we need to identify the frequency of the most frequent allele and the less frequent allele. So this is the minor allele frequency. In the majority of occasions, this minor allele is related to the disease, but not needed. It's not mandatory. And this is the effect. The beta in a linear regression normally is the statistical that is used. And this is the coefficient of beta that explains reflects the effect of that allele, the standard error and the p-value. And I highlighted this because this is that we will see now that will be used to compute the polygenic risk score. Okay? So if I'm not wrong, the first genome-wide association study was done in 2005. We have hundreds, thousands of genome-wide association studies, and this is the catalog that you can look for your trait of interest if you have already done the whole genome-wide association study. In radiotherapy side effects, we have worked a lot doing whole genome-wide association studies because we started doing candidate genetic studies, meaning that inferring that a gene, for example, ATM, was important for radiotherapy side effects. So we thought that looking for variants in ATM, that is a candidate gene studies, was a good strategy to identify variants behind radiotherapy side effects. But then, with the appearance of whole genome-wide association study, non-biased study, we interrogate the entire genome. And we have SNPs within head and neck, in breast cancer, in fatigue, that is a very recent published study, with fatigue in prostate cancer. We have done meta-analysis in prostate, and here in Milano, you published this development a method for generating SNP interactions from polygenic risk score. We have a good number of whole genome-wide associations studies, and we will have more, especially thanks to REC1, in fact. Because here is missing lung. So what is polygenic risk score? One of the possible outcomes of whole genome-wide association study to compute this polygenic risk score, because from the GWAS, we have a long list of SNPs. So each of one, we have the effect. That was difficult to manage this long list. So a good idea is to add in this effect, to have one score. So this is a statistical score, is a sum and adding different alleles with the weights that were highlighted, were computed in a whole genome-wide association study. So one thing that is I wanted to give you an idea is that either the GWAS and the polygenic risk score are statistical associations, are not identifying causal variants, okay? But managing the polygenic risk score is easy and allow another kind of studies, okay? So the PRS is a weighted combination of multiple common genetic variants, each of one individual, contribute very modestly to an individual risk of a trait, disease, or treatment response. So adding, you have only one score, okay? And this is the way that many of you are already, I'm sure, already done. And the beta here is the weight that we saw in the table of the statistics of the GWAS. The X is the number of risk alleles that can have two alleles. So the risk allele can be 0 because we can have in the two positions the reference of the other allele that is not conferring genetic susceptibility, conferring any effect. We can have in one allele the no allele risk and the allele risk, so that was one, and we can have in the two chromosomes the two alleles, the two allele risk would be 2, okay? And then you add all the not needed here. There are different statistical approaches. So there are people that selected only those that are at this threshold that we show, but also test and others that are below this threshold. and use a different strategy as LASSO, LEDPRIT. Then they validate, is recommended to validate this PRS in a different cohort that the GWAS that was highlighted, that list of SNPs. And this PRS can be easily introduced in predictive models, and is another predictor of the disease that you are analyzing, you are evaluating, in our case, the radiotherapy side effects. Okay, and normally is used, the PRS normally is the standardized PRS, the Z-score, that is computed regarding to a control population. Okay, so this is a graphical, nice way to see. So the distribution of this standardized PRS is a normal distribution, having the majority of individuals population risk. The individuals going to this side, to left, maybe they are protected. And in the right side, maybe they are prone to develop the disease or the trait that you are studying. So it means that either in the two extremes is not 100% sure that develop that condition. You need another factors, non-genetic factors, epidemiological, demographics, and environmental, to have a more lifetime percentage of susceptibility. So this table is to summarize and to do the wrap up. So what GWAS PRS can tell us? Variants that is statistically associated with a trait. What they cannot tell us by themselves is whether the variant is causal. Okay. And another important thing is to the risk stratification. So they can give us a relative genetic risk compared with others. But the certainty, 100% alone using this GWAS and PRS, is not certain. You cannot say that that individual, because they have that PRS, will develop that disease or that side effect. So that was the. And for you, if you are more interested to know GWAS and polygenic risk scores, I have included this summary of different studies. Okay. So what we're doing in work package 4 in genomics markers, work package 4 in Tetris. We are now with Anton, you know already, Carmen Moreno, who is going to help us and support it in the experimental lab work. Jacqueline, you already, some of you already know her, was a prior postdoc of the group. Vicky Reyes is the radiation oncologist in charge of the clinical trials in breast cancer patients in the radiation oncology department. And Goretti was the project manager. So the objectives is estimate genetic markers of interest for to be used in the retrospective required modeling in Work Package 2 and to be included in the CE market software in the Aquilab tool, and the same biomarkers in the prototyping the digital twins, either with the required and the prospective that we are going to do. We are going to collect. Okay, and this is the idea of this graph. So the Work Package 4 will fit either the retrospective part of the Tetris and the prospective. So what genomic markers we are going to estimate? I have explained today to you the germline variation. Germline means that is inherited from the previous generation, and you have that variant, the SNPs, the PRS, the SNPs that form that PRS, you have that variant in all your cells of your body. And we will compute polygenic risk score for different basal comorbidities of the patients that can prone patients before radiotherapy to develop side effects. Pulmonary, lung, heart, cardiac, and secondary cancers. And we will also, and I would like to have a future educational seminar for talking about somatic variation, That means that it's not inherited from your dad or from your mom, but is occurring during the life of the cells. And we call that somatic variation. And there are this kind of somatic variation in blood that we call clonal hematopoiesis of the indeterminate potential that has been seen that can have a role to predict cardiac events. Okay. So now we are working and finishing this part of the work for developing PRS. That was a work initiated by Jacqueline and is finishing by Anton from the cohort of required breast cancer patients. In required, the patients or the whole cohorts that we had, breast, prostate, and lung, were genotyped using an assay that is called Oncology that has been quite used in different diseases, especially cancer, doing GWAS for cancer. And the real genotyping is meaning that you has a signal that variant was in that position is around 600,000 SNPs. And after inferring, you can have 60 million of SNPs. And doing according to the hypothesis that you are or the question that you're interrogating, you do filterings, and you have less or than 60 million of SNPs to be used in the study. But the whole, let's say, after different kind of imputations based on different reference panels, is around 60 million of SNPs. So then we selected published SNPs. For that, first, we have different meetings in Tetris to select the comorbidities that are related with the pulmonary, cardiac, and secondary malignancies, and identify which comorbidities we could look for the PRS to include in the predictive models. So then, once we selected the different conditions, the different baseline conditions, we look for if was a GWAS of that condition, and if we had already a published PRS, and if that PRS was not only once validated, but was validated in different studies, and what was the performance. If it was really more than 0.6 of accuracy, so we selected that PRS as a good one, okay? Because you have, for diabetes, for hypertension, you have different combinations of SNPs and different PRSs, okay? So then we go to this is the list of 21 conditions, and we went to the PGS catalog. So we had a GWAS catalog, and this is the PGS catalog that you can find the PRS, the list of SNPs and the weights that you can take for computing the PRS if you have the variants. So this is a table that we initially, with Jacqueline, we were struggling to identify all the SNPs, but now we have learned, and now we have around most of the 21, I think all 21 of PRS has more than 70% of the SNPs in the required. So we have a confidence that the PRS is good. And we have already computed that for each of the breast cancer patients. And this is a summary of one of the PRS of coronary artery disease. This is the raw PRS, so the raw data. And the reference is in rows, in pink, and the required, the breast cancer patients, is this. Then doing a standardized Z-score in 3 different ways. So you normalize the PRS, and this normalized PRS can be used now in the predictive models. So we have computed this. We need to select one of these 3. One of these. They're going to take the 3 different normalized PRS on one, but this is the algorithm that is bioinformatically gives you different options to show you, okay? And ongoing, as Anton told you yesterday, we are thinking, because to have 21 PRS And for those phenotypes, for example, for those phenotypes that we have around 70 of the SNPs, if we, for diseases that there are different combination of SNPs, if we multiply, if we integrate these different PRS for the same condition, maybe we gain SNPs, and we gain predictivity. And another option is different related traits can be integrated in one PRS, and also you can gain a prediction value. Okay, for example, in this for cardiac disease, the body mass index, the lipids formation in blood, for example, and that PRS is maybe gaining more value and have more predictive role in the predictive models. Okay. This is ongoing parallel work, because also we had a conversation with Miguel that for the TWAS, maybe this integration is not, but in the models, we can have, I guess we were talking, maybe in the models in Work Package 2, we can test one type of PRS and then substitute with another PRS, but in a model that have the individual PRS, have the signal of the RNAs, for example. Okay, I don't know if I explained myself. Yes. So this is the future work. We need to do this for the retrospective required to look for somatic variants in a set of 75 genes that we know that somatic variants are related with myelodysplastic syndromes and cardiac complications. And we need to sequence, we need to select the cases and controls. We need to get the DNA from Lester, and we will do that also for the prospect. Okay. This is for finishing, and this is the Hereditary Cancer Genetics Group photo from this year. And we have 2 units. One is the clinic. So the whole group is coordinated by a medical oncologist, Judith Balmaña, who is specialized in familiar cancer. And I also have research projects in hereditary cancer, breast and ovarian cancer, inherited. So, and I am in church, I am the leader of the laboratory unit, and the medical oncologist is leading the clinical group. Okay. So this is the lab. This is a photo from the lab. These two, Cetare and Laia, are working in hereditary breast and ovarian cancer families, genetic susceptibility to this kind of hereditary cancer. And Carmen and Anton are working in radiogenomics project. And this is Jacqueline that was started the Tetris part of our work, and now is Anton and Carmen that we are working in hematopoiesis, in clonal hematopoiesis, the genetic variant. And this is the time of acknowledgement. So many people, the partners in Requite, the partners in RadPrecise, the partners in Tetris that you will see, also the radiation oncology in Vall, they are very willing and keen to participate always. The bioinformatics unit and statistical unit in VHIO, and also Tetris project, because we are now here thanks to Tetris. And that's it. Thank you very much.
That was great.
Thank you.
Thank you. Questions from the audience? Miguel is already. Maybe you I mean, you can repeat.
Or maybe we can use the microphone.
Because this is when the new line.
Maybe you can say the question here.
Well, one question about the PRS normalization. I have a question, because every time we add a new patient, you have to normalize again all the patients, or
It required, of course, because no, it's not a problem. will be for the next one, for the prospective. Let's say I imagine when we have the PRS for computing the PRS. For one patient, I would say yes, we will need to compute, right, Anton?
I'm thinking in the Apple software.
Yes, that is an issue we have to solve if we would like to go to the real clinic. When you would like to propose something to the clinics, you cannot think that every patient is of them. So either that normalization means that the mean that you then either we somehow can guess that means that standard deviation from required is the one we would like to use for breast cancer patients. So this means that you will normalize new patients, but using that mean and that standard deviation, so that is feasible.
Yes, yeah.
And I think that a single patient is not changing.
No.
But even 200 patients probably are not changing the 2,000 that led to the. So probably we have to discuss this.
Yes.
But for sure, in the future, the new patient comes with his SNPs measured, her PRS computed from the weights, maybe the meta PRS with all the kind of PRS computed, error value, and then if we have to standardize that, and I can see the value of standardization, we should say
there is an example that we can have. Well, I think it's we can go to have a very well established risk that is for breast cancer. And there is a software, similar software, that maybe we could, for the next, I don't know when, but we could have a demonstration of the use of this software for computing cancer, breast cancer risk. They are implemented for ovarian and prostate and so on. It's called CanRisk, and is now getting popular in the clinical units of family cancer. And they have behind the scenes, let's say, so you introduce the raw score, and then based on a value of the control population, the normalized Z-score is automatically computed and then added to the alcohol intaking, preconception pills, and a series of different non-genetic factors. And then the value, the PRS value, and then gives you the risk for that individual having all these conditions computed. But behind the scenes, this PRS is normalized. Yes.
Normalized in clustering?
Yes, yes. They have maybe, I'm not sure, maybe I made it up, but they have based on because that score has been work on different thousands cohorts that are in a consortium work in SIMBA, that is stand for Consortium of Modifiers of Breast Cancer, whatever, and thousands of controls. They have this polygenic risk score computed for thousands of controls. Yes, so that we could go for in that direction. Yes. Thank you, Miguel.
I have another question about the combination of PRS. Probably you will have some SNPs in one PRS and in another PRS. So after do the combination, because probably you have shared SNPs in
Yes.
different.
I would say hopefully you have.
It will be redundant. Some of the will be redundant, and we will gain some SNPs. Yes,
But you will have one gene included in one PRS.
Yeah, that we need to control to decide which of the two will we use.
But that's basically why people claim that using the multipairs is better, because the software.
See.
So from what I understand, people who use the multipairs claim that by buying the pairs, you can minimize the overlap, the same SNP. So it gives a bigger weight to those SNPs that you don't have multiple.
You mix the SNPs with the data. You don't calculate the PRS individually and then mix them?
It depends.
Or you use only the SNPs in combination, so just take all the SNPs together? I don't understand.
There are several ways of. There's several softwares to do so, and we haven't chosen one because of that. Most of them start with the summary studies. So we've used it before. Some of them, they just mix it directly after. So it's
Depending on the software we are exploring, we are in discovery phase.
I think that this idea of combining the PRS, both thinking on different PRS associated with the same phenotype, the same trait, or different PRS associated with different traits but pointing to the same disease, cardiac, pulmonary, secondary cancer, is really of value for two reasons. One is that you are developing a method to have something meaningful from this combination. Ways that the same need probably in different areas with different betas, and I am adding the worst thing is when in one PRS the beta is below 0 and in the other is above 0. That is making me crazy. But, from a causal point of view, but then you said those are not causes, those are statistical association, and it happens. So, and that's why I think that developing a method that is taking into account everything, or at least most of these issues, and arriving at a final proposed possible solution is a big value, both from a research point of view, from educational training point of view, and from the point of view of then having those used in the clinics. You cannot propose to a clinician PRS to be computed and then used to guide some decisions. Therapy, having a sort of summary that is putting everything together much more effective. The second level is that, unfortunately, in Requite, I think we will not, you're doing that, you will not have the power to discriminate between PRS, as most cardiac conditions are just labeled as cardiac conditions. At least half of them are just cardiac condition. Only the other half has that was atrial fibrillation, that was coronary artery disease. There is no, so the events are not so many, first, but then second, in most cases, unfortunately, in the CRF, there was only the field a new cardiac condition, a new cardiac disease after the end of radiotherapy, and the answer was yes. And there was not the field, which one? So that is also making me crazy. The value we have on which is the cardiac condition is from those clinicians saying no, But then in the text field saying this patient had atrial fibrillation in 2020. And so there you have the information. The thing is that you have to go to the test, free test in different languages, in different jargons and everything, but then you have the information. But for most patients, for at least half of the patients, there is only cardiac. So this, you cannot test, it is associated with the PRS for cardiac, for coronary artery disease, for the PRS for atrial fibrillation. So better to have one general and test that.
So for that, that is an important aspect to bear in mind for the master thesis of Anton, because we were asking what as besides Tetris, because in fact Tetris is not asking if that PRS for cardiac disease is a good predictor. Is another factor that together with the others will be in a predictive model, either with in the word package 2, in a statistical, let's say, classical methodology, or in the digital tool. So for the thesis of Anton would be a challenge that we will need to face. But because we are asking in the master if this polygenic risk score multiple or integrated, it has a difference between considering separate PRS. But for the Tetris, in fact, to have the polygenic risk score is independently of that, because it's another predictor. So maybe it's better to have this predictor, this PRS, than the case report forms, because they have defects. You know what I mean? So maybe the PRS is more objective. It's giving more. Is if we the PRS has a high association and is well computed.
But it's only risk.
I'm sorry.
It's only risk.
It's risk. Yes.
It's
Yes.
not the disease.
Yes.
That's not
But together with the other.
Another issue that we will have the conversation later. It is not the risk, but it is an endpoint. Establishing that the possible issue with the endpoint. We are not asking for p-values or something like that. We know we have few events, but that the endpoint is general. It's just a cardiac condition, a new cardiac condition. That's why I was thinking that a multiple PRS, which is putting everything together and being a risk for a generic new cardiac condition, maybe associated with therapy, taking the load and the target classification and everything. But the endpoint is
Yes.
required. Unfortunately, that is required where you have the data. The other codes are really indeed trying to specify the codes, so not to be generic, but required was generic.
Okay.
I think, are there questions from online? No one.
No. There is questions from the online audience.
So you can say goodbye.
So thank you very much for attending the online, and we close the seminar. Thank you very much.
Thank you.
Bye.
Bye.